It's a pleasure to be here, and I want to thank all of you for coming out on this blustery evening. I have just a couple of opening comments. First of all, a lot of times what you hear here is, well, we're going to present the latest research. Well, this isn't the latest research. This is a bit of a history lesson, because when these genetic technologies became available about 25 years ago, there was a lot of skepticism that they could provide anything useful for conservation. How is a DNA sequence going to change how sea turtles survive and persist? And so this body of research, having the technology available for the first time, we ended up answering a lot of questions about sea turtles in a fairly short period of time. And I hope you'll see that there are very definitely conservation applications to the work I'm going to present tonight. The other comment I wanted to make is, let's keep it informal. It's an intimate gathering. You can stop and ask questions anytime you want. I'm from Hawaii Institute of Marine Biology, which is a lab on a beautiful 27-acre island. If you haven't been out there, please, you can get on our website and arrange a visit. Come see us. Come see the shark tanks. Come see where all this science is happening. Oh, and of course, I want to thank Sea Grant for making all of this happen. This is going to be a talk in 4 parts. First, get into the natural history of these organisms, and then discuss the natal homing theory, the idea that turtles return to nest on the beach where they were born. Then a little bit of evolutionary history about this ancient group, and finally, tracking migrations with DNA barcodes and tracking turtles into situations that are hazardous for them, so we can tell which turtles are getting hurt in fisheries and other human practices in the ocean. So this is one of my favorite topics down here, the turtle. And that's Thaisi Sanchez, a volunteer for Tamar in Brazil, which is a great conservation organization. Sea turtles are fairly popular, so much so that all 7 species have postage stamps, multiple postage stamps in some cases. But all I want to tell you here is that there's 2 taxonomic families. There's the leatherback turtle in its own family. It's 100 million years apart from the other sea turtles. And then the other 6 turtles, well, I'll introduce all of them here in a moment. Life history characteristics of marine turtles. They have some commonalities, even though the whole group is over 100 million years old. They're globally distributed, mostly in the tropics. Females lay eggs on sandy beaches. You probably know that. The hatchlings emerge and swim off into offshore waters. And for the longest time, nobody knew what happened after that. Juveniles spend a few years, maybe up to 10 years in oceanic habitat, and then they switch to bottom feeding near shore. So these animals basically have 2 juvenile phases that are quite distinct. One out in the ocean and then near shore. Males, mature adults migrate to the nesting areas, and sea turtles live a long time. There's some pretty good data that they can go out past 100 years. In some cases, it's thought that they may take over 30 years to maturity and then breed and nest for another 70, 80 years. And it may even go further than that. Okay, this is a leatherback turtle. This thing has been reliably weighed at over 2,000 pounds, a ton. It can get 8 feet long. It can dive over 1 kilometer deep, which is 5/8 of a mile. So these things are competitive with the great whales in terms of diving ability. They feed on soft-bodied animals, jellyfish kind of things, and they've been observed feeding above the Arctic Circle in 0-degree water. Okay, now remember, this is a reptile. First thing you learned in school biology, reptiles are cold-blooded, right? Well, this animal isn't cold-blooded. This thing has learned how to partially control and elevate its body temperature. And it's got these mammalian features like bone that has blood vessels going through it. It's only in mammals. So it's an animal that over the last 100 million years ago has evolved halfway into being a mammal. Warm-blooded, amazing diving ability, so on. This is a green turtle, our beloved Hanu. And this animal is known for a couple of things. It's a herbivore as an adult, eats algae and seagrass and so forth, and it's also delicious. It has been the subject of the most intense harvest of any of the sea turtles. And for that reason, it's endangered in many parts of the world. This is a hawksbill turtle. This thing feeds on sponges. Now, if you know sponges, you know they don't have any armor. They don't have any protection. So how do they protect themselves? Well, they have what's called silica spicules, which is basically spines of glass inside their bodies, and most of them are toxic. So this animal, amazingly, makes a living by eating bags of toxic glass. Okay? It's also widely sought in parts of the world for its shell to make jewelry and other things. This is a loggerhead turtle. This gets more into the temperate zone. It's not strictly tropical, but it's called the loggerhead because it's got a habit of eating conch and other heavily shelled animals, which it can crush with that big musculature around the head. This is flatback. Not a whole lot is known about it because it only nests around Australia and New Guinea. And until recently, it was thought to be a green turtle, just a weird-looking green turtle. So this animal is ripe for a lot more study. The olive ridley is the most abundant, widespread turtles. There are nesting areas in Pacific Mexico and in India that probably host upwards of 1 million turtles. They're smallest, fast-growing turtle, and usually, like I said, usually abundant. Now, it's got a cousin. This is Lepida kelly's olivacea, the most common turtle. This is Lepida kelly's Kampai, the rarest of the sea turtles. This turtle only nests and feeds in the Atlantic around the Gulf of Mexico. It nests in one place in Mexico, just south of Texas, and it was known for, back in the 1940s, there was a film clip of this thing nesting, and it showed an estimated 40,000 turtles on the beach at once. They do this thing called arribada, which is Spanish for arrival. They mass off the beach, and then they all come ashore at once. And it's thought to be a predator saturation mechanism. There's just so many eggs that the predators are overwhelmed. They can't eat a real fraction of them. But this thing numbered at least 40,000 turtles in the 1940s. There was an unregulated egg harvest going up through the 1960s. And by the time the Mexican government figured it out, they'd been pretty depleted. But the Mexican government did the right thing. They put Marines on the beach and they protected that nesting beach. This whole species, one nesting beach. Unfortunately, at the same time, in the Gulf of Mexico, there was a U.S. shrimp fishery developing. And the shrimp trawls were drowning these things wholesale. So by the 1980s, late 1980s, there was only about 300 nesting females left. This thing went right to the brink of extinction. But through cooperation of the U.S. government and the Mexican government, they've managed to recover. They're now nesting in the low thousands. So it looks like this thing is well on the way to recovery. So that's our cast of characters. Let's go through a little of that life history. This is a green turtle, big 400-pounder, nesting at Ascension Island in the Atlantic. They'll come every 2, 3, 4 years. It's not a strictly regular cycle, but averaging 2 to 3 years, they'll swim from feeding grounds over to the nesting areas. The female will lay 3 to 7 nests of about 100 eggs each. Okay, that's a lot of investment. Okay, these females will drop 1/3 of their body weight, dropping eggs into the sand. Some 50, 60 days later, the little guys come boiling out of the sand, and they know that everything likes to eat them because they scramble for the water. And then they take off the beach. And as I said, that was where the scientific knowledge about turtles largely ended, up until 2 or 3 decades ago. It started to change with Archie Carr at University of Florida. He's more or less the godfather of modern sea turtle science. And his first innovations, he lived in cattle country in North Florida, where all the ranchers put zinc tags on the ears of their cows, their cattle. And he figured, well, I could put those zinc tags on sea turtles and find out where they're going, what they're doing. And he did, and he was very successful at it. Okay, here's one study site where Archie Carr had a student. Well, up at the top of your screen, you can see, what is it, Suriname? Yeah. Right. There's a nesting beach there, and there's a nesting beach out on Ascension Island in the middle of the Atlantic. And what the tags showed, that the turtles were feeding side by side along the coast of Brazil with extensive seagrass pastures. But one of the first facts that came out of this was that the turtles coming from Ascension to feed in Brazil would always go back to Ascension Island, never go back to Suriname. And this prompted Archie Carr to propose his famous natal homing hypothesis, that sea turtles, after a maturation period of 20, 30, 40 years, go back to the beach where they were born. They remember in a big ocean where they were born and go back and nest there. It's a charming hypothesis, but when I started this work, I thought it was pretty unlikely. Okay? Now, natal homing proved very difficult to test directly. You see that little hatchling there on top of mom? It's really hard to put a tag on that hatchling that's going to be there 30 years later when the female comes back to nest. Even if you could wait that long for your dissertation project. Right, Michael? Right, Melissa? Even if you could wait that long, there's a huge mortality on the hatchlings. It's estimated that only 1 in 1,000 makes it to adulthood. So it was really hard to prove. And that brings us to part 2, testing that natal homing theory. Mature females return to the same beach every nesting season. Is it the beach where they were born? Okay. Now, after Archie Carr proposed his natal homing hypothesis, another gentleman, Hendrickson, came up with a hypothesis that he called social facilitation. And he said, well, golly, I think that, when turtles get ready to nest for the first time, they just follow other turtles to a beach. And once they find that beach, they'll stick with it for the rest of their lives. Okay, that it was socially facilitated. The turtles just followed other turtles, found a good beach, and stayed there. Now, if you think about it, that explains why the tagging studies showed that adult turtles always go back to the same beach. And it's simpler because it doesn't require the turtles to remember where they came from 30 years ago. Okay? But these two hypotheses have very different expectations in terms of genetics. Okay? Imagine, if you will, a hatchling leaving rookery or nesting beach is the same thing. It goes through those post-pelagic stages that I told you about, juvenile stage, and then it ends up on an adult foraging ground. Now, if at that point, it just follows a female to any of the local nesting beaches, rookeries A, B, C, and D, then there's going to be a huge amount of interbreeding between those turtles. However, if it starts at rookery A and only goes back to rookery A, every one of those rookeries, every one of those nesting beaches will be genetically distinct. Okay? So this is where the state of the knowledge was on sea turtles, these two competing hypotheses. And I stumble into graduate school and I've got genetic tools that weren't available even 5 years earlier. and we can test this natal homing hypothesis. This is mitochondrial DNA, okay? It's a ring. It's in the mitochondria. If you remember your high school biology or your college biology, mitochondria are the batteries in the cells. They're the energy factories, okay? And they have their own DNA. And a key feature of it is it's only inherited through the eggs. It's only inherited from mom. Okay, sperm do not contribute mitochondrial DNA. So essentially, these mitochondrial DNA sequences are female inherited lineages. Why does that matter? Well, very little is known about the males because they don't come ashore. Okay? The females, you can tag them, you can poke them, prod them, study them on the beach, and it's pretty easy to do. Males don't come ashore. Nobody knows what they do, except there's ample evidence they act like males. That's two males on top there competing for the ladies' attention. Okay? All right, so let's get down to genetics. Haplotypes are different DNA barcodes. Here's Archie Carr's study site where the turtles are feeding shoulder to shoulder in Brazil, but always going back either to Suriname or to Ascension. We genotyped them, and 15 turtles from up there in the top Suriname had haplotype C. The 34 Ascension turtles had haplotype D, and one of them was one mutation different, haplotype E. Okay? Fixed diagnostic differences between these nesting beaches. Just dramatic evidence in support of natal homing. Lo and behold, I didn't believe it. They do it. They remember where they came from, and they go back there to nest. Okay? So we got this information and got a grant and surveyed turtles around the world, the green turtles. Okay? And the first thing we discovered was that green turtles don't nest in ugly places. This was really nice field work, okay? And we sampled Hawaii, and I'll get back to Hawaiian Hannam in a little bit. But now what about these other species of turtles? Now what you see at the top there is a map of Florida, and the pie charts indicate the genotype frequencies. And you can see that the genotype frequencies vary going down and up the Florida Peninsula. So it looks like, as best we can tell, they may not remember exactly the same patch of sand they came from, but they will come within a few tens of miles. Within, I would say, most of them make it within 10 miles of where they were born. And further research went out and checked this. The green turtle, we know. The Hawksbill turtle, the olive Ridley, the leatherbacks, they all do it. They all do this homing. Some are a little more precise than the others. Like the leatherback turtle can nest and then show up 100 miles away, but still it's in the same area. So all of these turtles are doing homing, as indicated by our DNA studies. Why does this matter? This is a leatherback turtle. We put in a poacher for scale so you could appreciate the size of it. What this means is that every nesting beach is an independent, what we call management unit. If a nesting beach is depleted or even wiped out, it's not going to be replenished anytime soon from turtles coming in from elsewhere. So this is, as I started, this is where genetics can show you really vital things. How do you manage sea turtles? Well, you manage them on nesting beach by nesting beach scale, okay? And this genetic conclusion is corroborated by Grand Cayman, the history of the turtle fishery there. Starting in the early 1600s, there was a fleet coming over from Europe to harvest green turtles. As I mentioned earlier, they're delicious. They're the original source of turtle soup, which was a delicacy in Europe. And Grand Cayman had probably over 1 million nesting females coming there at the time. So the boats came over from Europe year-round and relentlessly harvested these things. They'd stack them 6 high in the hold, and they could still be alive when they got back to Europe. Okay? By 1800, the nesting colony was gone. 200 years of unrelenting harvest, and the nesting colony was wiped clean. 200 years later, it hasn't come back. Okay? So the genetic data and the historical data tell the same story. Each nesting colony is a distinct management unit. And in some cases, that harvest can be substantial. You see what that is? Those are eggs. Okay, and if you want to talk about this later, I'd be happy to elaborate. But let's just quickly touch on the Hawaiian Hannam. Okay, in the upper left there is George Balas, the grandfather of sea turtles in Hawaii, sea turtle studies. He's been at it for 50 years. He was the guy in the 1960s that convinced, or early 70s, that convinced the state legislature to close the turtle fishery. Okay? He saw it coming. He was working with the turtle fishermen right here in Hawaii, and he told them, this isn't sustainable. You're wiping these things off the planet. And he's still active today. But what George discovered, among other things, is we know that hanu feed throughout the Hawaiian archipelago, but the map on the lower right shows the tracks of turtles that were satellite tagged. We could move from zinc tags now to satellite tags. Satellite tagged and tracked. And they all were going up to French Frigate Shoals, okay, which, as you can see in the top map, is about halfway up the archipelago. That's the major nesting site for the hanu in Hawaii. And as far as we can tell, it's a self-enclosed population. There's a few turtles that occasionally show up from outside Hawaii, but it's rare. Mostly, this is an isolated population that, again, is going to rise and fall on its own. And thanks to the protection that George Balas helped put into place, this population appears to be fairly recovered. It came back. When you've got animals that can lay 500 eggs in a season, they can bounce back pretty good. So 40, 45 years of protection, hanu is looking good. Of course, that opens up the issue of whether they could resume the fishery. And if you want to talk about that afterwards, I'd be happy to. Okay, moving right along. Part 3, just a little evolutionary history and how actually this evolutionary history can influence conservation of these animals. I told you earlier, the olive ridley, Lepida kelly's olivaceus, there on the right, is everywhere. It's all through most of the Pacific, although they're rare here. They're more coastal animals. The Indian Ocean, they got into the Atlantic probably a couple of 100,000 years ago, and they've colonized all the way up to Africa, South America, and they're starting to show up in the Caribbean. They also have big numbers in the East Pacific there, on the west coast of the Americas. Okay, Kemp's Ridley, found only in the Gulf of Mexico and limited in numbers. Now, they're recognized as distinct species, but the turtle biologists that work on freshwater turtles say they're not different species. Yeah, one's got a slightly more domed shell and one's a little different coloration, but we find that much difference in a single species in fresh water. So there was a lot of doubt about whether these species were legitimate. Why does this matter? This seemingly arcane scientific nomenclature. It matters because of things like the Endangered Species Act. If the Kemp's Ridley isn't a different species, it doesn't get protection under the Endangered Species Act. If the Kemp's Ridley is in fact just a North Atlantic version of the most common turtle in the world, then the Mexican government and the US government don't want to spend millions of dollars trying to hang on to one nesting population. Okay? So here's where this kind of arcane scientific naming stuff has a very direct application on conservation efforts. So what did we find? Well, this is what we call a phylogenetic tree. And on the top there, you can see the loggerhead. And the divergence between Atlantic and Pacific loggerheads, which were recognized as one species, was about a little less than 1%. Okay? The divergence between the olive Ridley and the Kemp's Ridley was twice that. This data showed that the Kemp's Ridley and the olive Ridley are about 3 million years apart. And this information, which with today's technology, a high school student could have put this together. Back then it was a lot harder and trickier, but this was used in Congress to justify the funding to save the Kemp's Ridley. Okay? Evolutionary history connected directly to funding to protect that highly endangered species.
And the Kemp's Ridley has also diverged?
No, that just showed the level of genetic diversity was in Kemp's. Yeah, at the bottom.
Got it.
Thank you, though.
And you have two subpopulations of Kemp's then?
Not two subpopulations. That I just put that in there
Oh,
to
just to say that that was Kemp's.
how much genetic diversity was within the Kemp's Ridley.
Okay.
I should have put in a comparable one for olive Ridley, but yeah, thank you. That's an excellent question. I should have pointed that out. Okay, let's move on to the green turtle, which was believed to be two species. Chelonia mitis through most of the range. And in the East Pacific, against the west coast of the Americas there's one that's smaller and darker, okay? And it was considered to be the black turtle, Chelonia agassii, okay? But this was also controversial. It's smaller and it's darker, but other than that, it's really not much different from the widespread green turtle. But the species status of the black turtle was being used to justify protection because it was pretty hard, battered pretty hard by collections like for leather factories and stuff. My buddy Naoki Kamizaki looked at this way back in 1995, said, if you look at the bottom quote, problematic to regard this East Pacific as a full species. He said the morphology just doesn't cut it. So we did the DNA. This is an old slide, but what I want to point out to you is there's two major branches in the green turtle, and they're at least 1 million years apart because these are tropical animals. And if you look at the map, it's hard for them to get between the Atlantic and Pacific Ocean. They cannot go around the bottom of South America, and only rarely can they go around the bottom of Africa. It's pretty cold there too. So the climate keeps them separate. But if you look at the top branch, it goes Australia, Japan, Hawaii, black turtle, Hawaii, and Oman. Okay? The black turtle, genetically extremely similar to Hawaii. So it looks like the black turtle is a colonist into the East Pacific probably since the last ice age, which would be 20,000 years ago. It doesn't look like it's a different species. Now, my colleagues and I, when we showed that the two Ridleys were different species, everybody gave us a pat on the back. Way to go, good job, it reinforces conservation. When we published this, we got a quite different treatment. Okay? I presented this information at a science meeting in Mexico, and I had a friend, Pedro, who I'd been working with for years, and he worked on the black turtle. Okay? And he said, no, it's different. We know it's different. It's different species. And I presented this data and said, okay, so Pedro and I disagree on this, but the important thing is that we can discuss it and we're still friends. Right, Pedro? Pedro stands up and says, no, we're not still friends. People got really heated about this. Okay, but it's science. You got to report what you see. Okay, so this went all the way into the top conservation journal of the day. There's the black turtle and the green turtle, and there was a big debate about it, but it ended up not being so harmful for the black turtle because it's an isolated population in the East Pacific. Okay, it's the East Pacific. If you don't know that area, If you've never gone surfing in San Diego, you won't know that it's cold. It's got cold water upwelling. So we think that the black coloration is just an adaptation to absorb sunlight for basking. That's all it is. A different color doesn't make it a different species. Okay? So the conservation of the black turtle continued, and it is doing better. It is recovering. Okay? Just a little of what we call biogeography and evolutionary history. Up there at the top, you have, well, let's start at the bottom. Okay? Blue is Atlantic, red is the Indo-Pacific, and green is both. And you can see with the leatherback turtle, the branches are all sort of mixed among themselves. This animal clearly gets between the Atlantic and Pacific frequently. The next one up is the green turtle. Look at that clean division between Atlantic and Pacific. And it's long enough, it's 1 million years. So it's long enough, you don't want to say they're different species, but they're definitely evolutionarily different below the species level. They're genetically quite different. Okay, then the flatback, we'll skip that. The hawksbill, the same thing. It's tropical, and it's almost completely genetically differentiated between Atlantic and Pacific. You see there in the red branch, there's one short blue twig that probably indicates the Indo-Pacific one. One of them snuck into the Atlantic recently. The loggerhead, which has no problem feeding around southern Africa, you can see the lineages are pretty well mixed up because it's a temperate animal and it can get into cooler water. So anyway, the point of this slide is that the climate and their temperature tolerance seems to be guiding the evolution of this group. Okay, and with that, we move into part 4, tracking migrations. Now you notice, you remember, Suriname and Ascension Island had diagnostic differences. They were genetically different with diagnostic genetic differences, which means you could take a drop of blood from a turtle feeding in Brazil and say, this is an Ascension turtle. This is a Suriname turtle. Okay? So hopefully you can see where this goes in tracking these turtles out in the open water where very little is known about them. Okay? Very often it's not as simple as a diagnostic difference between nesting colonies, but it's enough. There's genetic differences. Sometimes it's just the frequencies of the different genotypes, but it's enough. Okay, let's start with this loggerhead turtle. Off the west coast of Mexico, Baja California, in that area, there were known to be somewhere around 100,000 juvenile loggerhead turtles. Okay, we're talking the size range of 12 inches, 24, maybe 36 inches. Okay, juveniles, and lots of them. Okay, but the biogeographic mystery was, where are they coming from? There's no nesting beaches anywhere in the East Pacific. And I personally went down through Central America looking for this. Okay, because occasionally somebody would say there's loggerhead nesting, but it always turned out to be the Ridley, the olive Ridley. So there's no nesting in the East Pacific. Where are they coming from? Okay, the only nesting is in Japan and Australia. Okay, so a Japanese researcher put out a little note saying, well, you know, there's a Kuroshio current North Pacific gyre. I think those animals are coming from Japan. And there were physiologists, a couple of prominent physiologists in North America that scoffed at him, said that's impossible. That's a third of the planet. There ain't no animal leaving the beach this size that's going to cross a third of the planet. Okay? So we decided to try to test this. And to do it, we had to get turtles off of Baja California. And in the upwelling zone there, it's very nutrient rich. There's these things called red crab. They're actually shrimp. They're about this long. But they're so thick there that you can sort of see a red patch from space. There's dozens of miles long, continuous patches of these things, and the juvenile loggerheads were feeding on them. So we went out there with my Mexican colleague, Alberto Abreu, and we were going to collect little drops of blood from these guys. We had to go 30 miles offshore, and we were going with the local fishermen. So this means like a 20-foot dory, open boat, yeah, with a 20-year-old Johnson outboard motor, and there was no GPS, there was no cell phones, there was no radios, there was no nothing, okay? And we're going out there, and on the way out, we're stopping other fishermen that are coming in from fishing out there. And I noticed their boats are full of sharks. The sea turtles aren't the only things feeding out there. It's chock full of sharks. So we go out there. Oh, and you know, these boats that go out there to fish, every once in a while, one of them lands in Hawaii. Okay? They get blown offshore, and the lucky ones, I mean, some of you have seen this in the paper, I know. One of our longline fleet picks up a boat that's been at sea 100 days, and they've been living on rainwater and fish. Okay? And those are the lucky ones. They get found, right? Blown offshore in Mexico, they're at sea at 100 days in a small dory. And you know what? This was on my mind. I was really thinking about this. So also, we now know that there's sharks out there. So the protocol was, when we found a turtle basking on the surface, we'd cut the engine and glide up real quietly, and one of us would jump in the water, get the turtle, get it over the boat, get it in the boat, get the researcher in the boat before the sharks came, because they pretty much always came. As soon as we started splashing around in that water, sharks came. It was hairy. Silky sharks and some other species. And my collaborator, Alberto, he was about 100 pounds lighter than me. So we quickly decided he should do most of it, because we could get him out of the water a lot quicker. Okay, now while this is going on, our Hawaiian sea turtle guy, George Balas, phones me and says, "Hey, Brian, the longliners north of Hawaii, it's the darndest thing. They're picking up little baby loggerheads north of Hawaii." So we sampled the north of Hawaii, Japan, Australia, Baja California, and what did we get? Well, the Japanese beach was all genotype B and C. Australian beaches were all type A. And you can see a couple of A's in the drift net fishery. I'm sorry, I said longline, but it was drift net. And a couple of A's in the feeding ground, but the vast majority of them match Japan. Itaru Uchida, the Japanese researcher, was right. These things are coming from Japan, okay? The longest migration still to this day in the ocean known to man. Remarkable. Okay, now when we go to publish this, well, the reviewers say, yeah, but all you've demonstrated is that some turtles get blown offshore in Japan. You haven't, they're just strays. You haven't demonstrated that they go back to Japan to nest. So we put that arrow with a question mark under it. We said, well, maybe they're riding back on the equatorial currents. And we published it. And this guy, Antonio Resendez, was getting very excited about this. He was rehabilitating loggerhead turtles, cold-stun loggerhead turtles in Baja. And he was doing it in his swimming pool, and his wife didn't like it because they pooped a lot. But he had turtles that were, he had a turtle that was looking like real energetic, like pacing the tank like a tiger. And he slapped a tag on it, Rosita. He always named his turtles. He put a tag on Rosita and let it go. And lo and behold, 400 days later, Rosita's tag is recovered in Japan. So it demonstrated they weren't just strays coming over from Japan, they were going back. Okay? Now the sad part of that tale is, how did they recover Rosita's tag? She drowned in a fishing net within a couple of 100 miles of where she started life. So there's that. So Antonio Resendez is really excited. He gets some money for satellite tags. And he puts 2 satellite tags on these turtles. And did they go south and just meander with the equatorial currents? No. When they left Baja California, they went straight as an arrow for Japan. Dead on navigation. So we were wrong about that riding the equatorial currents. But that's science. You put out your best guess, and if you're wrong, that's okay because somebody made the effort to carry the work another step. Okay. Similar story in the Atlantic. It's getting late, so I'm going to skip this and go straight to the Hawksbills. It was a similar story in the Atlantic in that the Loggerhead Turtles are doing the same thing. They're going out to sea, and then they get all the way into the Mediterranean. Like loggerheads that originate in the Florida nesting beach can be caught in Italy, okay? But then they come back straight as an arrow. This is the Hawksbill turtle, valued for the jewelry it can make, those scales. If you could go to your grandfather's bureau and pull out an old pair of glasses that were tortoise shell, guess what those were made of? Before plastics, they were made from this guy. And it was lethal extraction, okay, which drove them way down. Now, in the 1990s, Cuba announced an intention to resume harvesting Hawksbill turtles. Okay, there's an international treaty, CITES, Convention on International Trade in Endangered Species, which banned all international trade. But Cuba was going to take an exemption, and they said it's because the turtles that are swimming around Cuba come from Cuban nesting beaches. So it's nobody's business whether we harvest them or not. In the language of international conservation, they claimed there was no range states. There were no other countries that would be influenced by this harvest. Now, after you just saw what loggerheads can do, you might be skeptical. But there was no data to contradict them, and this was an international scandal. They were going to start collecting this stuff and shipping it out primarily to Japan, which has a centuries-old craft. I mean, beautiful artwork that's made from these shells. Okay? All right. Now, I'm just going to keep this nice and simple. Going down in that table, going down the column on the far left, those are the rookeries, the source populations. Cuba, Puerto Rico, Virgin Islands, Antigua, Bahamas, Costa Rica, Belize, thank you, and Yucatan. Okay, those are the potential source nesting populations. And we sampled in countries throughout the Caribbean, Cuba, Bahamas, Dominican Republic, Puerto Rico, and Virgin Islands. Now just direct your attention to the column under Cuba, okay? It says 45% of the turtles feeding around Cuba are indeed from Cuba, but 19% are from Puerto Rico, 21% are from the Virgin Islands, and down at the bottom, 10% of them are from Yucatan, okay? So this data showed you harvest in Cuba, you're going to be affecting countries throughout the Caribbean. To put it more graphically, when we sampled in the U.S. Virgin Islands, in Puerto Rico, we found turtles from Belize, Mexico, Cuba, Puerto Rico, and U.S. Virgin Islands. Now, to look at it a different way, how about that Mexican nesting colony? Where did those turtles show up? Well, we found them in the Bahamas, Cuba, Puerto Rico, and the Dominican Republic. Okay? So, I would like to say that this data stopped the Cuban harvest. It was used to stop it, but it was really the public outcry, the international outrage that they were going to start harvesting such an endangered species. So to conclude, only 2 minutes late. Okay. To conclude, the genetics shows that every nesting beach is a distinct isolated management unit that's going to rise and fall on its own. If it's over-harvested, it has to recover on its own. But you know, you don't need a PhD in genetics to figure out that you shouldn't be harvesting the eggs on the nesting beach. And you shouldn't be harvesting females on the nesting beach. If you cut into that critical reproductive spot, I mean, anybody can figure that out. Don't harvest, over-harvest. Here, it's in the open ocean where this genetic data is starting to make a real difference. We can show if it's a dredging operation or a long line or drift net. We can say, okay, that's taking individuals from a highly endangered nesting colony. Or the converse, we can say it's taking animals from a huge nesting colony, so it's not much of a problem, okay? But it's in the great unknown of the open ocean where genetics shows which nesting populations are being impacted, okay? And with that, thanks.
Questions for Dr. Brian Bowen? Raise your hand.
Bill.
Yeah, in the mating process, do the males, or has it been determined that the males actually are offshore on the nesting beaches, or does that take place somewhere remotely? And how long before the eggs are fertile so they can be laid?
That's a great question, and it's still not fully resolved. But the males do aggregate off the nesting beaches. But
so it's pretty immediate then, the fertilization. Yeah,
but they have to fertilize them before that shell calcifies. The egg has to be fertilized before the egg gets hard. Of course, you know, mechanics. Sperm bumping against an eggshell. Okay? But it's also known, I mean, this is phenomenal. At the Grand Caymans, they had a turtle farm because their nesting colony had been wiped out. And there's a case of a female that was in a pond for 2 years, crawled up on the grass, dug a hole, and laid viable eggs. So they can store sperm for a long time. Okay?
Is there no possibility that they're parthenogenic, that sea turtles, that they can reproduce without males?
The question is, can they reproduce parthenogenically, which is asexually, which we now know some sharks can do.
And lizards.
And lizards.
Dogs.
And fish. To the best of our knowledge, no. They can't reproduce parthenogenically. They can't get rid of us males yet.
At least the turtles, right?
Yeah, you're no help. Jeff?
Do they imprint on the sand and the smells, or is it magnetic?
Thank you. That's a great question. How do they do this incredible trick? Okay, there's a lot of theories that they smell it, but the one that's come to prominence that is almost certainly correct is they have magnetite in their head, which is the same stuff birds use to migrate. And so this magnetic compass tells them not only direction, but they can sense latitude and longitude by the angle from the North Pole. They can tell how far north they are. So that's an interesting question, but it brings up another point that I told you these guys have been around for 100 million years, right? And they've been nesting, and they were doing fine until recently. If they still nested at the same nesting colonies they nested at 100 million years ago, they'd be extinct. Because 100 million years ago, the Antarctic was warm. Okay? Continents drift, climates change. And so natal homing cannot be perfect. Perfect natal homing is a recipe for extinction. And we know now, there's in the big nesting colony in the Caribbean in Costa Rica, out of 35,000 turtles that have been tagged, 2 have been shown to relocate major league. We call them gravid waifs, crazy ladies. But they occasionally, it's rare, but occasionally they colonize new nesting beaches. And if you think about it, they have to. Okay? Beaches come and go. What happens when the beach disappears? You know, you're going to go extinct, or you're going to find somewhere else. So, thank you. That was, yeah.
What are you working on sea turtles these days? And if you are, what are you working on specifically that you can share?
I haven't worked on sea turtles in 7 years. I work on reef fishes now. And I'll come back and tell you the story about the origins of Hawaiian reef fishes, where they came from. That's one of my current projects. But what's happened is, as I mentioned, 20, 25 years ago, there was a lot of skepticism, and wildlife agencies like U.S. Fish and Wildlife, they didn't want to give money for genetics. What's genetics going to tell us? Okay, so these were basically demonstration projects. And now they have a multi-million-dollar program to do this. So I sort of let the big dogs do it. I had my fun, and I started it, and I'm proud of that, but it's now a big government program. Hi.
So you had a picture of the 2 male turtles and a female. So can multiple males fertilize a female or one?
The question is, can multiple males fertilize a single female? With a couple of exceptions, we've used DNA fingerprinting to look at the eggs, see how many dads are in the nest. With a couple of exceptions, they all have multiple dads. But part of that is because the males are really aggressive. I mean, the males have these big spikes on their front flippers where they dig into the females. And you can always tell a recently mated female because she's still bleeding from here. But the males are so aggressive. In some parts of the world, fishermen will throw a barrel in the water. And the male will climb up on the barrel, and they just reel it in. Bang. That's how they harvest turtles. And in some parts of the world, I can testify, the males have come after unfortunate snorkelers. Yeah, it's not pretty, but I mentioned there's a big nesting colony in South Florida, right? Well, what's south of South Florida? No, Miami Beach. There's a big nesting colony, summer nesting colony, next to one of the most famous beaches in the world where there's tons of people in the water. And almost every year there's one or two of those unfortunate interactions. Yes.
Other than Maui for 35 years, and I just noticed in the last year that I think a lot of the turtles have lesions. You know, these big, huge teeth. I'm just guessing. But also, I've noticed that there are certain turtles that are coming onto different parts of the North Shore that never used to come here so much.
Oh, okay. Well, part one, the lumps are tumors. Fibropapilloma, it's related to the herpes virus. And it's a mysterious thing because it started showing up in green turtles and loggerhead turtles in Hawaii and Florida almost simultaneously in the late 1970s. Figured that out. And it has since spread pretty much to every ocean basin. Okay? There was a time in the 80s and 90s where we were really worried about it because up to half of the turtles, at least around Oahu, had these tumors. The incidence has actually started to go down. It looks like they're just starting to develop immunity to these things because they can be lethal. They can be lethal. And as to the turtles you have coming that are showing up on the North Shore, those are our Kailua Bay turtles because the sand in Kailua has extended out after a few storms in the last 5 years, and it's covered the algae. So whereas I used to be able to sit in water this deep, stand, I could watch Hanu come up, take a breath right there in front of me. I haven't seen a Hanu in that bay in 2 years. So now I know where they went. Now, they'll shift habitats. If they find that there's not enough food here, but there's good algae up on the North Shore, they're pretty good about figuring that kind of thing out.
Brian, I think there's some connection between the fibropapillomatosis and invasive algae in some cases. I've heard that might explain the sort of simultaneous nature of it.
But that's disputed.
Yeah.
There's some vets that argue pretty strongly against it, but the correlation's definitely there. The invasive algae showed up, and these turtles started getting tumors. But it's a herpes virus. Did it come in with the algae? Yeah, it's still not settled exactly what's going on there. But, hi.
Sorry. You mentioned the crazy lady turtles or whatever. So ones that go off, and relocate not to their original nesting. So do you know if those turtles that are like born there and hatch there, do they come back to that spot and it's new nesting?
We think they do.
Could you repeat your question?
Oh, okay. Those crazy females that move to a new nesting beach, do their progeny come back to the new nesting beach? Okay. It's if the homing was genetically hardwired to go back always to the same beach, then like I said, they'd be extinct. So we believe, and knowing that they use the magnetic tissue, we believe it's imprinting when they're either in the nest or when they're leaving the nest as hatchlings. So nobody's done that experiment yet, okay? Because again, it would take you 30 years. But our belief is that the progeny of the crazy lady would go back to the new nesting beach. It's a good point, yeah.
Is there an effort to I guess, mimic crazy ladies and relocate nests to beaches that have been depleted?
Yes, you're nailing them. That's a great question. Oh, stay near the mic for the folks out there in Radio Land. The question is, has there been an attempt to transfer eggs to a new nesting beach and see if they'll come back to that new nesting beach? And actually, the answer is yes. I misspoke a little bit in answering your question. When the Kemp's Ridley got down to about 300 nesting turtles, this is disaster. This is imminent extinction. There was an attempt to start a nesting beach in Texas, okay, which is just about 100 miles north of the natural nesting beach on Padre Island, actually, the barrier island. And they incubated the eggs in these metal huts to protect them from predators, raccoons and such. And then they let them go. But there was an obscure little piece of literature, scientific literature. This is such a great lesson that in a journal you never heard of called Copia that said turtles have temperature-dependent sex determination. And what happened was, if the eggs are incubated a little, just 2 degrees warmer than normal, they're all males. For the first 7 years in Texas, they produced only males. And males ain't going to help you nesting, right? So they lowered the temperature so they got more females, and they figured they got it fixed. But what they didn't know was the magnetic orientation that they were using. And they were being raised in metal huts. So they released them, and they start nesting. I think the furthest north was North Carolina. They were nesting in Florida, but now some of them are nesting in Texas. So provisionally, the program was a success, and they moved them, and at least a few of them came back to the new nesting beach. But as one of the wildlife researchers said, when they got reports of these turtles nesting in Florida, they said, well, that's pretty good homing for a turtle raised in a bucket.